I have to confess before I start that the aim was to talk about methodological issues and predominantly about statistical parts. This was my understanding. And I'm a clinician. I understand nothing about statistics. Don't ask me any questions on statistics, please. Not that you can ask me questions about clinics either. I left clinics some time ago, so. I'm charged with talking about methodological issues and relating to patient selection and a CHMP reflection paper. It is not a guidance, it's a reflection paper talking about or reflecting on their experiences in the first few years of pharmacogenomics. So this is slightly dated, if you will permit, in the sense that the paper was put out for consultation about 10 months ago, and we are in the process of reviewing it. So I will take you through some of the thoughts which went through our minds at the time when we looked at the data sets. There obviously are updates to this, and hopefully you will find that I'm not saying anything completely controversial or contradictory. I've got to be careful about this. This is going too fast for me. Right. What I'm proposing to do is to talk about a little bit of the background in personal medicine and regulation, and why do we need a guidance or reflection paper, and what does the paper say, and what next? The flow of the slides will be smoother when I get used to the mouse. As you see, one of the things that our observations was that biomarkers represent an additional test burden. And as you saw, that they can be quite disruptive. They raise too many questions for anyone to give a comfortable answer. And no one is comfortable giving an answer to many of those questions. The markers need qualification validation, and most importantly, in my mind, they need the context. And why, when, and how you want to replace the old practice of looking at a patient's face and deciding this is the best, most appropriate therapy for them to say, oh no, I've got to depend on some tests. They also need some reliability and discriminatory ability and need cutoff points. And when we were looking at the data sets which CHMP had access to when this paper was conceived, we found that some of these areas were quite debatable. They come up, whenever you look at biomarkers, patients, and regulation, we tend to get a set of questions which are very commonly asked. And how do we qualify and validate? I don't propose to answer that question. What level of evidence is required for a biomarker, a pharmacogenomic biomarker, in regulatory terms? And do we need randomized control trials for qualifying every marker and identifying its benefit or its role? And when will we accept retrospective data? And can we use them as surrogate endpoints? As you would have gathered, I don't intend to show that I have answers to any of those questions, and I will only air some of the thoughts, and that's the reason why I'm reading them out, so that if I stop, I might forget the next one. In my mind, the regulatory objective should be, of any clinical trial or data set, is the evaluation and recommendation needs to be scientifically robust. Hopefully will stand the test of time, not necessarily for years, but at least some time. It has to be clinically relevant and is practical to implement. I don't propose to say that there are a number of markers. I mean, you will have a number of markers which you can identify and classify, use subgroup patients. But does it help in order to streamline the therapy, or is it clinically relevant? This brings us back to the rare diseases discussion, which you already heard before. How do you make a consistent group, a consistent effect to follow based on one single marker or a set of markers? Of course, that is just a statement which I think which everyone has heard, and I don't really need to repeat it. Apologies for the typos in the first thing. What we need with the biomarkers is to have a clearly demonstrable difference between groups, whichever way you want to classify them as. If you take, for example, marker negative and marker positive populations, in the left-hand panel here is show that virtually there is no difference. Never mind the fact that some people are carriers, or some people are marker positive. If you have a predictive marker, I would expect that there is a clear difference in the treatment effect. And this is what the reflection paper was primarily concerned about, and not about prognostic value, more of predictive effect and predictive value. Based on the experiences of CHMP, the number of products have in their label some information about the pharmacogenomics incorporated. It's not much different to the one which my colleague Michael Pakanowski presented earlier, but some of them are a little bit old. The question now comes in as to what were our learnings from some of these. I won't go into details of each of those products, but this spans the entire period between 2000 to 2009, I think. We haven't taken any more on this. I've restricted myself to what the paper talks about. Just to give you a short overview of what the reflection paper is about, the aims were to identify products where genomic biomarkers influence the development or raised questions, and to identify and streamline some regulatory expectations, and to discuss the CHMP experience and concerns. It is restricted primarily to those products which came through the centralized marketing authorization process, or some of the major safety issues. It's not a detailed review of literature or of our experience or the methodologies used. I think I really don't need to go into the details of these. And this is the standard all-comer test that you would use, you would test or do not test the treatment effect based on a marker. And such a design we would expect that would be adopted where the results of the testing is not readily available at multiple clinical sites. And we have seen that the science has progressed. But this is the situation where you still have an early marker, or a marker in early stages of development, where it is insufficient to inform a randomization. I seem to have lost control of the mouse. Please, could I have the next one? Thank you. If you were to look back at one of the data sets that we saw, the one which comes to mind first is the unselected markers for unselected design for EGFR mutations for Geftinib. You will see that there are a fair number of marker positive patients and a fair number of marker negative patients. And you would think that based on those small sets of patients with lung cancer, you would be able to make some decision process. But if you actually compare it to the bottom panel here, bottom thing here, you'll see that the proportion is fairly small in comparison to the overall population size. The point of interest that I have I was going to show is that if you look at the green lines, there is a clear difference between marker negative and a marker positive population. But what strikes me most is the fact that the marker negative population seem to do worse. Is that a true finding? One would only be able to say if all of these people were sampled for DNA analysis and mutations. And unfortunately, we haven't got that information. So it sort of limits us. It limits us to our conclusions as to whether that difference is clear. And emphasizes the point which was made before, that we would like to have biomarker data or DNA samples from majority of the subjects in a clinical trial, even in an unselected design. Unsurprisingly, the next example isn't any better, or at least it emphasizes the point. We've heard about KRAS story and about its multiple markers, And whilst the defense at the time of evaluation of the data was that it's a prospectively defined retrospective analysis, what became evident subsequently was that whether this is a prognostic marker or a predictive marker, a combination of the two, we, at the time, for those who are not aware of the history of panitumumab vector-based regulatory aspects in Europe, it was first granted what is called a conditional authorization, which meant that they have to review their process periodically, and the product needed to be renewed. And at the second renewal, it was suggested that based on the clinical data generated, that mutated KRAS may be detrimental if you use Vectibix, implying that it is a marker of safety, not just a marker of efficacy, as we would have thought, in terms of just if you were to administer it to wild-type people with wild-type KRAS. What became evident subsequently was the fact that there is a further relationship for KRAS mutations with BRAF and RAS and PIK3. Unfortunately, at the time of the regulatory submission, these were not defined. And how do you incorporate data which arises from biobanking to the knowledge that you have at the time of regulatory authorization? In my mind, it's a difficult one. And I think it will be very, very important to seek some clarity on what one can do with that sort of data generation. Do we simply take that the regulatory process takes its time, and you will decide based on the information available within the dossier, or would you be guided and permit more information to come in? Science would dictate that you would take more information, but that, of course, is a CHMP decision. It isn't for me to comment. But it is an interesting point to raise in terms of designing clinical trials. The issue I'm trying to address is that if these informations were in the public domain prior to a design of a clinical trial was thought through, could you have done any better, or could we have done any better in terms of incorporating more newer markers? I don't know the answer to that. The second design which we tend to frequently see is enriched and targeted design. I think one of the questions which we had in the first session was that how do we select out, or what amount of information do we need in the early phases to select an enriched or a targeted design? I won't go into the design details. You're all experts at it much more than I am. I was going to two examples as to what issues will arise. I've lost the control of the mouse again. Okay, I'll have to live with the slides the way it is. The question which comes up for enriched studies is that it's quite probably easier to decide you will do an enriched design study if we have the hope and expectation that you will find a good treatment response. Herceptin, everyone knows. The point which I think we'd have to bear in mind is that if it takes about 30% of all breast cancers are Herceptin-2 positive based on IHC, we would have to remember that IHC staining is not infallible. And there was 20% discordance between the clinical trial assay and those which are available currently. To complicate matters, there have been reports that there is a response to Herceptin even in those who are seemingly Herceptin negative. Puts anyone into a difficult situation as to whether you want to include marker negative subjects at all in the development program or in a clinical trial, or you're happy with just following the enriched design. From a regulatory perspective, you might be excluding a large patient population who we don't know the actual effect is. There might be benefits. Contrast it with vemurafenib in melanoma, you will see that majority of the patients with melanoma have V600 BRAF mutation. And if you exclude 4 or 5% of the subjects, it might not be such a big issue. It isn't an easy question to answer whether to include marker negative or marker positive patients, is all my point is. And that at that point of the reflection paper, it talks about the difficulties. Oh, not again. No. Can you move the slide forward, please? Thank you. The next design, which is not part of a regulatory submission, but what I thought would be interesting to show is that it answers some of the limitations of unselected or enriched designs, but it clearly has an implication for sample size and expense. As a regulator, I would like to see, ideally, a trial of this nature for any number of markers that you can use. I live in hope that we will see more of these trials on a regular basis. What the observations of the clinical trials at the time was that predictive values need to be generated in the patient population that reflect anticipated clinical use, and that the enriched design does not always validate the marker or define its utility in a broad population. It will depend on its predictive value. I won't go into those statistical details, and I'm sure my colleagues will talk later, will talk about it. The issue here would be that are we sufficiently confident with the data that we've generated that this is enough for us to progress this from a regulatory submission to clinical practice? Could you move that one for me, please? Okay. Just a few more slides on talking about a different aspect, just restricting myself to clinical trials. What sort of data would you expect to be generated in the safety markers? Here are 3 examples in which we have the difficulties, so the slide is self-explanatory. And for abacavir, as we all know, that there was a prospective study, and there is replication, the clear evidence. Whereas when the events are rare, such as with carbamazepine, it's almost impossible to expect a prospective trial. Most of my regulatory colleagues will be very horrified at the fact that I'm standing up here and making a statement such as that it is impossible to conduct a prospective trial, but that's my personal feeling, that it will be difficult in order to do a prospective trial in such a situation when there is a clear evidence of harm. But of course, there are discussions about which racial group or which group it will apply to. We need to find a way of addressing those issues. Warfarin has been around in use, is probably as old as I am. And you will note that there is virtually no replication so far, and everyone has found a different level of importance for either of those 2 markers that I have not put on the slide. Of course, there are other markers which you will find even more complicating in the sense of the association. What we do not have in terms of warfarin is that whilst there is evidence of association, there isn't any clear indication of how to alter the dose or what the maintenance dose should be. And of course, this is part of the European EU PAC trial, and we hope that you will get some information based on that, and the SPC the label will be modified based on that. Okay. Here is another example of the predictive ability, but this is, some of you might recognize this. It showed, whilst a very good relation with association to a particular marker, it was not universally applicable. And the CHMP, when they were looking at it, was not convinced screening patients had sufficiently reduced the risk of hepatotoxicity. An old example of population predictive ability is going back to carbamazepine story, that we think that we have a very high level of importance in Han Chinese and the Thai patients, but it doesn't really apply to HLA-B 1502 will not apply to the European, Caucasians, or Japanese populations, where a different level of risk and a different markers is possible. So what do we think are of retrospective data analyses? Our experience has been that it is often based on associations, and that it suffers from what is called the winner's curse. The first result is usually the best, and you'll never be able to replicate it. And it often overestimates the effect size. And the follow-up studies are often underpowered, and the phenotype heterogeneity is not taken into account. There is also a level of bias, which the subsequent speakers are more qualified to talk than I am. Based on all our experience, there are a few caveats which the reflection paper talks about, is that you would expect for any retrospective data analysis that data should be from a well-conducted RCT, and that the biomarker status should be available from all or majority of the subjects. And there should be a prospectively stated hypothesis, and there should be replication. The difference between marker positive and marker negative patients should be clear, or as clear as one can get. So, what is the experience at CHMP? That it's a lot of post hoc analyses, and that there are a number of open questions. As you will see, that I have only answers to some of them. And your next question, I anticipate, is that can we use adaptive designs to overcome some of these questions? And you can use different strategies for adaptive designs in my mind, and that is dependent on the availability of the marker. If it's available early, you take a certain scheme, you adopt a certain scheme, and if it is not available, you adopt a different scheme. Actually, on second thoughts, I should stop commenting here, and you can ask Martin Posch, who will talk next. Thank you very much.